Welcome to our new video on anatomy of flowering plants. Once upon a time, deep in the heart of the forest, there lived a group of plants. These plants were all different, each with their own unique characteristics and abilities. Some were tall and majestic, reaching for the sky, while others were small and delicate, nestled close to the ground. As the plants grew, they discovered that they had a secret world inside of them, a world made up of tiny cells. These cells were organized into groups, called tissues, each with their own specific functions. Some of the cells were able to divide, while others were not. The cells that could divide were special, they were called meristematic tissues. They were found in certain regions of the plants, such as the tips of the roots and shoots, and were responsible for the plant's growth. These special cells were able to create new cells, which would then become permanent tissues. The permanent tissues, on the other hand, were made up of cells that could no longer divide. They were divided into two groups, simple and complex. Simple tissues were made up of only one type of cell, such as parenchyma, parenchyma, and sclerenchyma. These cells had different shapes and functions, and played important roles in the plant's survival. Parenchyma cells, for example, were responsible for photosynthesis and storage. They were found in most of the plant's organs and had thin walls made of cellulose. Parenchyma cells, on the other hand, provided mechanical support to the growing parts of the plant, such as young stems and petioles. They were found in layers below the epidermis of most dicotyledonous plants and had thickened corners due to the presence of cellulose, hemicellulose, and pectin. Sclerenchyma cells, on the other hand, were thick-walled, dead cells that provided support and protection. They could be either fibers or sclerids, and were commonly found in the fruit walls of nuts, the pulp of fruits like guava, pear, and sapota, and the seed coats of many plants. Complex tissues, on the other hand, were made up of many different types of cells. These tissues were responsible for the plant's more advanced functions and were found in specific regions of the plant, such as the roots, stems, and leaves. As the plants grew, they adapted to the diverse environments around them, using their different tissues to survive and thrive. And so, the plants and their secret world of cells lived happily ever after, in perfect harmony with the forest. In the land of plants, there were three different types of tissue systems that worked together to keep the plants strong and healthy. The first system was the epidermal tissue system, which protected the plant and helped it absorb water and nutrients. The second system was the ground tissue system, made up of simple tissues like parenchyma, parenchyma, and sclerenchyma, which helped to support the plant and give it structure. The third system was the vascular tissue system, made up of complex tissues like the xylem and phloem, which helped to transport water and nutrients throughout the plant. These three systems worked together to keep the plant alive and healthy, but they were not all created equal. The epidermal tissue system was the first line of defense, protecting the plant from outside threats. The ground tissue system provided structure and support, while the vascular tissue system was responsible for transporting vital nutrients and water throughout the plant. As the plant grew, it began to divide into two different types, dicotyledonous and monocotyledonous plants. Dicotyledonous plants had roots, stems, and leaves that were organized in a specific way, with an outer layer of epiplema, a middle layer of cortex, and an inner layer of pith. Monocotyledonous plants, on the other hand, had roots that were similar to dicotyledonous plants, but they had more xylem bundles and a larger pith. Their stem had a protective layer of epidermis, a cortex that consisted of multiple layers, and a pith that was well developed. As the plant continued to grow, the dicotyledonous plant began to undergo secondary growth, which allowed it to form new xylem and phloem tissues. The monocotyledonous plant, however, did not undergo secondary growth, and so it remained the same size. Despite their differences, both types of plants were able to thrive and flourish in their own unique ways, thanks to the hard work of their three tissue systems. The epidermal, ground, and vascular tissue systems worked together to keep the plant healthy, and allowed it to grow and prosper in the land of plants. As the plant grew, they went through two types of growth, primary and secondary growth. The growth of the roots and stems in length with the help of apical meristem was called primary growth. However, as the trees grew older, they also increased in girth, and this increase was called secondary growth. The tissues involved in secondary growth were the two lateral meristems, vascular cambium and cork cambium. The vascular cambium was responsible for cutting off the vascular tissues, xylem and phloem. In young stems, it was present in patches as a single layer between the xylem and phloem. Later, it formed a complete ring. The cambial ring became active and began to cut off new cells, both towards the inner and outer sides. The cells cut off towards the pith matured into secondary xylem, and the cells cut off towards the periphery matured into secondary phloem. The cambium was generally more active on the inner side, which resulted in the amount of secondary xylem produced being more than secondary phloem. In temperate regions, the activity of cambium was under the control of many physiological and environmental factors. In the spring, the cambium was very active and produced a large number of xylary elements with wider cavities. The wood formed during this season was called spring wood or early wood. In the winter, the cambium was less active and formed fewer xylary elements with narrow vessels, and this wood was called autumn wood or late wood. As the stem continued to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layers got broken and needed to be replaced to provide new protective cell layers. Hence, another meristematic tissue called cork cambium or phellogen developed, usually in the cortex region. Phellogen was a couple of layers thick and cut off cells on both sides, with the outer cells differentiating into cork and the inner cells differentiating into secondary cortex. The cork was impervious to water due to suberin deposition in the cell wall. The cells of secondary cortex were parenchymatous. The plants that had undergone secondary growth were known for their hard, durable and resistant heartwood, which was dark brown due to the deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layers of the stem. This region comprised dead elements with highly lignified walls and was called heartwood. It did not conduct water but provided mechanical support to the stem. The peripheral region of the secondary xylem, known as sapwood, was lighter in color and was involved in the conduction of water and minerals from root to leaf. As the seasons changed, the plants continued to grow and adapt to the changing conditions. The bark that was formed early in the season was called early or soft bark, and the bark that was formed towards the end of the season was called late or hard bark. And so, the plants stood tall, providing for the forest and all its inhabitants for many, many years. The end. This is a summary of the chapter Anatomy of Flowering Plants from Class 11th NCERT Biology book. We hope you enjoyed our video on Anatomy of Flowering Plants. We hope the story-based format helped you to understand the chapter better. If you have any questions or feedback, please reach out to us. Thank you for joining us and stay tuned for more exciting content.